Hello guys, I want to say welcome to our YouTube class and thank you for being part of what we are doing here. I will still enjoy you to share our video to as many that you know will need it, not just within the ASUT family. You can still share to other students in UNN, UNIZIK, Photo, EBSU, and so on and so forth. And even all over the world, as many that you know will benefit from this video because it's advisable. I want to encourage you to send to them. So today we are going to be discussing something very important, something very profound, and that is the science of genetics. The science of genetics. The science of genetics. Just a minute. So the science of genetics is also important. As a matter of fact, this field of biology or this field is central uh, to biology, occupies a central position in the study of biology because it involves both plants and animals, and it also involves the discrete units that is transmitted from parents to their offspring, from generation to generation, and this includes all the organism that you can call a living thing. So there is no organism that you can call a living thing that does not have the discrete units or the hereditary units, which is called gene, that we are going to be studying in genetics. By the way, genetics is defined as a study of gene or the study of the behavior of gene. Some people can also say, okay, genetics is the study of heredity and variation. And of course, when you talk about heredity, you're talking about the transfer of traits, characters from parents to their offspring, from generation to generation. Then when you talk about variation, you're talking about the differences in traits that exist among organisms. So when you study or when you're studying heredity or you're studying variation, what you're studying is genetics, because genetics can be defined as a study of heredity and variation. And like I said, in the simplest term, you can also say that genetics is the study of gene or the behavior of genes. There is a man that is central to the study of genetics, and that man is somebody that will always be mentioned when it comes to the study of genetics, and that is the Gregor John Mendel, Johann Mendel, or Gregor Mendel. Gregor Mendel was the one who discovered the fundamental units of heredity. Even though when he discovered this, uh, it was not widely accepted within the scientific community at that time, and he went back to the monastery. But then he made a statement or a comment, "Meine Zeit wird schon kommen," meaning that my time, that was in German language, meaning that my time will surely come. And his time actually came in the 1900, when three scientists working, in the same area he worked in, he used the Sioux sativum That was the plant he used in his experiments. So three scientists performed similar experiments and they got the same results. That is uh, Erich von Skemeg and his colleagues. So when they got that result, it now resonated with the result that Mendel got before he went back to monastery. And that is to show that his time actually came. Hmm? That is to show, just a minute. That is to show, sorry, that his time, I hope you can see the board. That is to show that his time actually came when these three scientists performed this experiment. I said Erich von Sesenec, Erich von Sesenec. These are foreign names. They'll have the one they call the concurrence and the Hugo de Varies. Hugo de Varies later made a discovery. Hugo de Varies, Erich von, Erich von Sesenec, then concurrence. These were the three scientists that performed similar experiments and they got similar results that Mendel got before he went back to the monastery. So that is a brief history of what genetics is all about. But then I didn't go into the full history. I think if it's possible, I may make a separate video where I will give a full outline of the history, where people like Aristotle made their postulations. Charles Darwin also made his own postulation. Where we mention the theory of epigenesis, the theory of preformationism, the theory of pangenesis. All these theories are found within the historical view of genetics or within the historical development of this subject that is called genetics. But I want to leave that aside. It's something that one can read and understand. So if we have time, I can also do that. But let's go into the main aspect of genetics, which is important and a bit technical. Now, I want to start by saying that cells have chromosomes, and each of these chromosomes is this in pair. Of course, within the autosome, we have 22 pairs. Then the sex chromosome has one pair. So that will give us total of 23 pairs of chromosomes that are found within the cell. And each of these chromosomes, like I said, is this in pairs. One coming from the father and one coming from the father and one coming from the mother. And because this chromosome is this in pair, they are said to be diploid. Hmm? They are said to be diploid. It's said to have a diploid condition. It's said to have a minute. So, like I said, this chromosome state where each chromosome is represented twice is said to be a diploid condition. It's a chromosome state where each chromosome is represented twice. Like yes, it's represented twice. Like this is chromosome. This can be chromosome one. Then we can have another set of chromosome, another set of chromosome. This is chromosome two, and this is how it is until we get to all the chromosomes we have within the autosomes. Okay. Uh we'll now look at I want to be taking some of the points, genetic points, one after the other. Hmm? I want us to be looking at it one after the other. And before I go into the next point, I think I mentioned this in my last video. I want to repeat it again. Members of this pair of chromosome, members of this pair of chromosome, or these two members of this chromosome, chromosome 1, these two members are said to be homologous. They are homologous chromosome. So this chromosome and this chromosome are said to be non-homologous. This one are non-homologous because they are members of separate pairs of chromosome. But members of this pair of chromosome, so this and this, are said to be homologous. Homologous. Then this and this are said to be sister chromatids. These are sister chromatids. Sister chromatids. Chromatids. Then this and this are non-sister chromatids. Non-sister chromatids. So these are some of the technical things. So like I said, this and this are sister chromatids. This and this are sister chromatids. This and this are non-sister chromatids. Then this and this, the whole of this and the whole of this are non-homologous chromosomes. Then this and this are homologous chromosomes because they are the members of a particular type of chromosome. Type in the sense that we'll have chromosome 1, chromosome 2, chromosome 3, and the rest of them. Okay, then it is important you also note that chromosomes carry different sets of units. Chromosomes carry different sets of units, and these different sets of units are responsible for heredity. They're responsible for heredity. Chromosomes carry different sets of units. They carry different sets of units, and these sets of units are said are responsible for heredity, and they are called gene. So let me repeat what I said before. I said chromosomes carry different sets of units, and these sets of units are responsible for heredity, and they are called gene. Mendel called it discrete units, and they are responsible for the resemblances you see between parents and offspring. So Mendel said that there is something that is religiously handed over to man by God, or handed over to man by God, which of course parents transmit to their offspring from generation to generation. And that thing is called the gene. That thing is called the gene. So we can also call it discrete units. So I said chromosome carry different sets of units, and these sets of units are responsible for heredity, and they are called gene. That means that, and it's also important to know that these sets of units occupy definite physical position on the chromosome. Yes, these genes we are talking about occupy definite physical location. On the chromosome. There's a location within the chromosome where they occupy, or there's a position within the chromosome that they occupy. And that physical location is called locus. That physical location is called locus. If there are many, you can call it loci. If there are many, you can call it loci. Locus, they will have loci. Okay. Then these the genes can be in alternative forms. The genes that are located on this physical location can be in alternative forms. And the alternative forms of gene is now what we call allelomorph, or alleles, the alleles. This alternative form of gene is what we call the alternative form of gene is what we call what? Allelomorph or alleles. Allelomorph or alleles. Allelomorph or alleles. So let me give you an instance. For instance, if we have a gene here, gene A, gene B, we have gene A, or have gene C, gene D, then we can simply say that A and D are alleles. Alleles. Why are they alleles? Because they occupy the same physical location. Then and capital B and capital A and capital B are also alleles. Why? Because they occupy the same physical location. Then C and A, then D and capital B are known as non-alleles. Non-alleles. Why are they non-alleles? Because they occupy different physical location on the chromosome. So I hope that is understood. And it's important you also notice that or know that alleles may control the same character. Alleles may control the same character. But even though alleles control the same character, like we have this C and D as alleles, but even though they are controlling the same character, they may of course have different information or may contain different information. That means that if the character that is being controlled by this alternative form of genes is height, for instance, if it is height, then they may contain different information in the sense that this is capital T and this is small letter t. So why this one is tall, then one is short. And this will also bring us to what is called homozygous and heterozygous. When the alleles of a gene pair are the same, when the alleles of a gene pair are the same, then it is said to be homozygous. And the organism containing such allele is said to be homozygote for that particular character. But when the alleles of a gene pair are different, then it is said to be heterozygous, and the organism containing such allele is now said for that particular character. I know what I'm saying, for that particular character is now said to be a heterozygote. So what it means is that this allele will have allele A, they contain the same information. They contain the same information in this case, but as you will have allele T and allele small letter t, they actually different because one is capital T and the other is small letter t. So in this case now, they are said to be heterozygote. They are said to be heterozygote. The members of this allele contain different information, just like initially said, that members of alleles may contain different information, even though they are representing one character. So in this case, the information is different. And in the case of heterozygous, or in the case of a heterozygote organism, you will now discover that one allele mask, the word mask, eclipses, covers, hides. I'm trying to use so many words to explain this mask. So in the case of heterozygous, one allele masks or covers the expression of the other allele. The one that masks the expression of the other allele is said to be dominant. Then the one sorry, that is the allele that is masked or that is suppressed from expressing itself is now said to be recessive. So we have dominant, we have recessive. We have dominant, we have recessive. So the one that mask the expression of the other is said to be dominant. The one that is suppressed or that is masked from being expressed is not said to be recessive. A minute. Okay, so we are going gradually. Let's now talk about what other important concepts. Of course, you also know what is called a genotype. A genotype, the sum of genes contained by an organism, is called a genotype, and the measurable. Uh, uh, just a minute. So the measurable characteristics or distinctive feature of an organism is now what we call the phenotype. The measurable characteristics. The measurable characteristics. Some people thought that phenotype is only outward. Okay, you're looking at an individual physically, and that is what we call phenotype. No, it's not just that. Phenotype goes beyond the physical expression you see on an individual's body. We are still going to look at that in details. So, but for now, note that a phenotype, for instance, if you get somebody's blood group and it is A or O, of course, that is a kind of phenotypic expression. You are not looking at the blood type physically of the person's body. So some people think phenotype will be color of the hair, sorry, the texture of the hair or the color of the hair or the skin. That is the only one they understand as phenotype. So the totality of measurable characteristics, even if it's physiological or morphological, is what we refer to as phenotype. So phenotype is also phenotype P is a product of G plus E, is a product of G plus E. That means that the environment can also influence the genotype. This is gene plus environment. We are still going to look at this when we get to quantitative and qualitative characters. We are going to see how that the genotype influences, how that the genotype, yes, how that the phenotype, sorry, is influenced by the gene and the environment. So phenotype is a product of the gene and the environment. The next one we're going to be looking at now is the genetic symbols. Genetic symbols. Now, in the study of genetics, there are symbols that are used to denote some certain important terms, and it is important we understand, we note or we understand these symbols. Symbols. For instance, when we say P1, we mean first parent, first parent, first parent. When we say P2, we mean the second generation of parents. The P2, the first generation of parents, P1, second generation of parents, P2, and we have P3, P4, and so on and so forth. Then when we say F1, we mean the first filial generation, F1, first filial generation. Also have the second filial generation, and so on and so forth. Then when we use when we use square, for instance, it means a man. When we use circle, it means a woman. When we use circle, it means a woman. So in this case now, if we have this, it means a man that is normal. A man that is normal. But if we put a dot here, put a dot here, put a dot here, put a dot here, it means that this man is normal, but this dot is an indication that it is carrying or that he's carrying a, or is a carrier of a disease. Yes. Simply put, the presence of this dot is an indication that a man is a carrier of a disease. Then, if we now do something like this, then it means that this man is being affected by a disease. It also means that this woman is being affected by a disease. Then if we now put something like this, the same thing, and put cross, it means that the man has gone to the land of his silent fathers. That means that the man has died. Cross means that the person is dead. Cross means that the person is dead. Then if we have something like this, it means dizygotic twins. As against dizygotic zygotic twins. As against the twins that are identical. This one is called identical twins. When you begin to study the pedigree, the human pedigree. Okay, so having crisscross these elementary terms, because we are going to be meeting some of these elementary terms, and that was why I took this little time to explain some of these technical terms that are used in genetics. Let's now go into the work that Mendel did, the Mendelian laws. The Mendelian laws. Like I said, Mendel worked with sum sativum. So this is the pea plant, that Mendel worked with, the pea plant that he worked with, the sum sativum. And in the course of his working, in the course of his research, he found out so many things which were now organized into laws of Mendel. Those discoveries he made during his research were now organized into what we now call the Mendelian laws. The Mendelian laws. Mendelian laws. We have the first and the second law of Mendel, and we're going to be looking at the first law and look at some of the crosses he did, and also look at the second law. The first law of Mendel is also called the Monohybrid law, and the second law is called the Trihybrid law. So in the first law, according to him, when gametes are formed, each gamete receives one gene from each gene pair, one from the father and one from the mother. When gametes are formed, during the period of formation of gametes, that is gametogenesis, so each of the gametes will now receive one gene. Remember we said genes are always in pairs. They are always in pairs. And because they're always in pairs, each of the gametes will now receive one gene from the two pair, one coming from the father and one coming from the mother. So if one gamete receives the two and the other does not receive, is a case of abnormality. So that is what Mendel pointed out. And let's assume that we have a character, and that character is height. So in this case now, we have that the P1, the parent, this one is tall, this one is short. So this is the first parent. The first parent is, let me write this up. So we have P1, the first parent is tall. The first parent, let's assume this is the father. Of course, fathers are always tall. Then this is the mother, and mothers are always short. Now, in this case, now you bring out, this is G showing gametes. You bring out this T and you circle it to show that these are the gametes. These are the gametes. Then we'll have F1. And let's see what happens in the case of F1. In the case of F1, we now discover that we have T go to T, we give you capital T, small letter t. And that reminds me, remember that this parent is homozygously tall. Hmm? Homozygously tall. When the alleles of a pair of a gene are the same, remember I said this before. When the alleles of a pair of a gene are the same, they are the same, they're said to be homozygous. But when they're different, they're said to be heterozygous. So this man is homozygously tall. This woman is also homozygously short. Homozygously tall, homozygously short. So in the crossing now, we'll have this going to this, we'll also have this going to that's capital T, this. They'll have this capital T, this. We'll have this, that's capital T. Sorry, let me start all over again. So I have this. So T, capital T small letter 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 T. Hmm? So what's the implication? The implication is that Mendel discovered that this was actually the Mendel's crossing. You know, he crossed so many plants and so many varieties of these plants based on different characters. Mm. He checked the color, he checked the height and the color. As a matter of fact, he crossed the purple flowered plant and the white flowered plant. We're still going to come to that, but we're using height as one of the traits or characters that he worked on. So this is an indication that Mendel discovered that when he crossed a tall plant with a short plant, he discovered that all of the offsprings were tall, and Mendel was confused and surprised. How is it that I cross a tall plant with a short plant? And I'm now seeing in the first village generation that all the members were tall. What now happened to the short gene? Or what now happened to the short plant? Was it that this gene, disappeared? Or what now happened to it? And because of a curiosity, he now said, okay, let me uh, self-cross this Fourier generation, this first Fourier generation. So Mendel now came, and this will give you P2 and cross this plant and self-cross the plant. Eh? This is G2. If you are showing that it's gametes, then you have to cross. That is a sign to show that it's a gamete. So when you now cross, this is now F2. You now found that T, capital T, then that is this, capital T, then T, small letter t, then capital T, small letter t, then small letter t, small letter t. So this was what Mendel found out. He now found out that the character he thought disappeared in the first Fourier generation resurfaced again. And you can see it clearly stated here. And this will now give us ratio of ratio of 1 to 3, that is 3 is to 1. 3 is to 1. So you can see this explains the first law of Mendel. The first law of Mendel. During the formation of gametes, each gamete will receive one gene. Each gamete will receive one gene from each gene pair. Each gamete will receive one gene from each gene pair. One from the father, one from the mother. So this is a gene pair. This is a gene pair. You can see at the end, you see this is one from this, one from this. Each gamete will receive one gene from each gene pair, one from the father and one from the mother. One from the mother and one from the father. And this will now give us what we call the segregation ratio of 3:1. So 3:1 is the segregation ratio. So Mendel, at the end of this experiment, found out that three of the plants out of the whole planet across, 3 out of 4 were tall, while one was short. So the gene he thought disappeared resurfaced again, but that was in the second filial generation. So this explains the first law of Mendel. So even if we use the white color flower, the pea plant, and the purple color flower pea plant, it will still give us the same thing because it follows the same principle. It follows the same principle. So let's now go into the second law of Mendel. The second law of Mendel, which is also called the dihybrid law. Now, in the second law of Mendel, he was trying to say something that members of different pairs of alleles are sorted independently into gametes. Members of different pairs of alleles. So, these different pairs of alleles, independently on their own, going together, they are sorted independently into gametes. In other words, he's trying to say that the segregation of the alleles of the first gene occurs independent of the segregation of the alleles of the second gene. That was what Mendel was trying to explain to us. And he now used two characters instead of the one character or the single character he used in the monohybrid law. So in this case, he now checked this is P, now use a tall plant, then with white color, and sorry, with purple color, this is purple, tall, and cross it with a short plant with white color. This G, uh, this G, are we going to use P? All right, let's use P now. Let's use P. Let's use P, even though you see the same thing. So this was what he did. Just a minute. So this was what he did. Mendel now cross a tall plant with purple flower, then a short plant with white flower. So this is white, this is white, this is tall. Then we now have gametes. We now have the gamete, which is T. Just pick one of these, T, then P, then T, then P. This will now give us this and this, T, then this and this, P. So this will now give us the first filial generation, the first filial generation having capital T, small letter t, capital P, small letter p, capital T, small letter t, and capital B, small letter p. So this is the first filial generation, Then to get the offsprings for the second filial generation, then what we now do is to step cross this first filial generation, just like we did in that of monohybrid law. We now step cross the first filial generation. So let's separate it well. This is capital letter T, then this, this. So if we cross, we now have this going to this, will give us capital T, capital P, then capital T, small p, the small letter T, capital P, small letter T, capital P. So if we use this, we can use it to construct what we now call a Punnett square. We can use this now, this information, to construct what we now call a Punnett square. A Punnett square. One, two, three, four. 1, 2, 3, 4. Okay, so we have this to be here. Then we have this here. We have this here. Sorry. This capital P. Yes, and small letter this P. So also have it here. Let's also have it here. Let's also have it here. Let's also have it here. So if we add this, we now get capital T, capital T, capital P, capital P. And what does it mean? It means that the plant is tall and has purple color. Then we have here capital T, then capital P, capital P. So you see the same thing. We also have tall and purple. Tall and purple. Why? Because this P is dominant, while this small letter P is recessive. So what you see C is tall and purple. Then this and this will give you capital T, small letter T, then capital P, it will still give us tall and purple. Tall and purple. Then this and this, capital T, capital P, small letter T, small letter P. Sorry. I hope I don't make any mistake here. Sorry. So this capital T, small letter T, capital P, small letter P. This is still tall and purple. Then for this, we have capital T, then capital P, small letter P. This is still tall and purple. Then we have this capital T, small letter P. So this one now means tall. But instead of having purple color, it now has white color. Because the small letter P, like we said, signifies the white flowered plants. Then we have this now, capital T, small letter T, capital P, small letter P. This is tall and purple. Tall and purple. Then this is capital letter T, small letter T, P. This is tall and white. Tall and white. Then here we have capital T, small letter T, capital P. Capital P. So this is tall and purple. Another tall and purple. Remember, if you have capital T, small letter T, it's still tall because the capital T which is the dominant, is masking the effect of the small letter t. Then we have capital T, small letter t, capital P, small letter p. This is still tall and purple. Then we have small letter t, then capital P, capital P. So this is short. First time short is appearing. This is short and purple. Short and purple. Then we have capital small letter t, capital P, small letter p. This is short and purple. This is short and purple. Then lastly, we have capital T, small letter t, then capital P, small letter p. This is tall. This is tall and purple. The capital T, small letter t, small letter p, small letter p. So this is tall and white. Remember, anywhere we see small letter p, we call it white. Then we have small letter t, then capital P, small letter p. So this is short, and short and white. Sorry, short and purple. This is short and purple. Short and purple. Then have small letter t, then small letter p, So this is short and white. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we're gonna have at the end of the day, we're gonna have 9 is to 3 is to 3 is to 3 is to 1. 9 is to 3 is to 3 is to 1 is what we call the dihybrid segregation ratio. The dihybrid segregation ratio will give us 9 is to 3 is to 3 is to 1. 9 is to 3 is to 3 is to 1. All right. So the whole of tall purple, that was what I counted. 1, 2, 3, 4, 5, 6, 7, 8, 9. The whole of this tall purple give 9. They will now have tall and white. 1, 2, 3. Tall and white is the first 3. Then what again? We now have short purple. 1, 2, 3. Short purple 3. Then short white 1. So can you see? This is the Punnett square and how we construct the offsprings based on the 2 different characters that we have crossed. And these 2 different characters are tall and purple. That means the plant is tall and has a purple flower, and another plant that is short. Sorry, I see where I wrote tall here. I meant to write short here when I was explaining this. Please, it's a mistake. But sometimes you'll be saying another thing and your hand will be writing another thing. So what I meant here, I just looked at it now, and that's why I wrote tall. This is not tall, it shouldn't be tall. This is short, then this is white, then this is tall. Of course, I've explained that the one that is a capital letter is a tall one. All right, so this is the hybrid crossing. Let's now talk about trihybrid crossing. Trihybrid. I remember. Okay, let me. In the trihybrid crossing, we are not going to get the uh, F2 offsprings. A minute. So, in the trihybrid crossing, the trihybrid crossing, we are not going to get the F2 offsprings. This board will not accommodate it. That will be giving us around there. This one gave us 16. 9 plus 9 plus 9. That is 7 plus 9. That should be 16. 16 F2 offsprings. Then that of trihybrid will be giving us 64. 64. 64. So we are not going to go into that. This board cannot accommodate that. And I don't think it's too necessary for us to do that. I will just tell us how similar Mendelian principle applied in that of monohybrid and the dihybrid is also applied in the case of trihybrid. We even have polyhybrid crossing. But in this case, instead of going through this manual method, we now use a formula. We're still going to come to that. So for trihybrid crossing, we have a plant that is tall. That plant has purple flower, and that plant has black pod. Let's use black pod, black pod, black pod. Which color are we going to use now? Just a minute. So let's use anyone. Let's use black pod and green pod. Black and green pod. Black and green. So this one is short, having a white color and having green pod. So if this is F1, then we'll have the gametes T, P, G, P, G, P. Then F1 offsprings, 1, bring this, bring this, bring this, bring this. Sorry, this should be capital. This should be capital. So bring this, then this is this. This is capital P. Capital P, then G, then small letter G. So what you now do is to self-cross, just like we did before. You self-cross. What we want to do is to get the number of gametes that will now produce the offsprings. That will now produce the F2 offsprings. The number of gametes that will now produce the F2 offsprings. And we're going to get about 8 gametes. In the dihybrid crossing, we got about 4 different gametes after self-crossing. So I'm going to self-cross this now. And after self-crossing, we're going to get 8 different gametes. 8 different gametes. The number of F2 offsprings in that of the hybrid crossing was 16. 4 times 4, that is 16. So in this case now, we're going to get 8. 8 times 8 will give us what? 64. 64. So we are likely going to get 64 different F2 offsprings. 64 different F2 offsprings. Just a minute. Okay, so let's cross T. Let's expand this. Let's expand this. Okay, we can see the without expanding. So T goes with a P. T, capital T with capital P goes with capital G. That is number one, the first gamete. Number two, we're using the same T. We're using the same T, P again. Instead of this G, we'll go with this small g. That is 2. Let's go to 3. 3, we still have capital T, then go with small p, then capital G, then 4, we go with same capital T, then small p, then small g. Then number 5, we'll now start with the small t because we are done using the capital T to cross others. So now I have small t, yeah, capital P, capital G, 6, we have small t, capital P, and yes, so small t, capital PG, small t, capital P, small g. Then number 7, we have small t, small p, capital G. Number 8, which is the last one, we have small t, we have small p, we have small g. So these are the 8 different gametes. These are the 8 different gametes that will now produce the 64 F2 offsprings. 64 F2 offsprings. Like I said, if you use the same method, of course you can do this at home. You are going to get the same result, and you are going to see F2 offsprings. All right, for polyhybrid crossing, is so much that it's not something you can do. So what scientists now did was to formulate uh, to bring a formula, or to produce a formula that can now be used to get the number of gametes and the number of F2 offsprings. Our interest has always been to get the number of gametes and the number of F2 offsprings. So in this case now, if you are given the number of characters, then you can now use it to get the number of gametes. The formula is 2 raised to power n. So assuming the gametes, the characters are 4, 2n raised to power 4, which is 2, let's say 2, okay, 2 raised to power 2 times 2 times 2 times 2. Actually, 2 times 2, 4, 4 times 2, 8, 8 times 2, 16. That should give us that should give us 16 F2. Sorry, that should give us n. This is the formula for getting the number of gametes. That should give us 16 number of gametes. But if we have uh, if we have if we have if we have okay, let's use the same thing, the same 4 characters, but then the F2 offsprings is 3n, 3n, 3n. So in this case now, to get the F2 offsprings for this, you now say 3 times 3 times 3 times 3. So by the time you multiply it, whatever it gives you will now become the number of F2 offsprings. Remember, number of gametes is 2n, number of F2 offsprings is 3n. Okay, that should give us around 81 if we are to multiply it. That should give us around 81. So if the number of genes under consideration and the number of characters are 4, then it will give you 16 gametes, then it will give you 81 F2 offsprings. What's the time? Okay. All right. I think, okay, let's quickly look at another thing. Let's quickly look at another thing. Let's quickly look at another thing. So, this experiment that Mendel performed, or the researches that he conducted, uh, uh, how do I want to put it now? The researches were not uh, so accurate, or let me say 100% accurate, because other scientists now began to perform researches with respect to this, and those researches led to what we now call modern genetics. And that was when they saw some defects in some of the principles or some of the laws of Mendel. And some of those things they discovered could not be explained using Mendelian laws. And this can be called exception to Mendelian laws or development of Mendelism. You know, Mendel has laid the foundation, and we applauded him for that. But then some of the things he said, or some of the principles he brought, could not be used to explain some certain phenomenon or phenomena in genetics. And that is what we want to discuss now. Some of the developments of Mendelism. One of them is what we call the uh, codominance. 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 Well, let's start with incomplete dominance first before we go into codominance. Incomplete development of Mendelism. Hmm? The first one is incomplete dominance. And in the case of incomplete dominance, what happens? In the case of incomplete dominance, you now discover that some alleles lack the dominant and recessive characteristics in such a way that each of the alleles are capable of expressing themselves to a certain level. So, like I said, in this case, incomplete dominance, you now discover, or scientists now discovered, that some alleles lack these dominant and recessive characteristics, and some of them are not capable of expressing themselves to a certain extent within the heterozygous. Yes, within the heterozygote condition, they are kind of capable of expressing themselves. So what I'm trying to say is that in the case of incomplete dominance, the dominant allele, the dominant allele does not influence complete masking of the effect of the recessive allele. The dominant allele does not completely shield, cover, or mask the effect of the expression of the recessive allele in such a way that the intermediates have sorry the intermediates have separate character, or they have a distinct phenotype different from the two parents. So an example of this is what happens in the case of Mirabilis jalapa. Mirabilis jalapa is a 4 o'clock plant. Mirabilis jalapa. Mirabilis jalapa is a 4 o'clock plant. We call it 4 o'clock plant. In this plant, you discover that there is no, uh, just a minute, let me plug. So sorry, I needed to plug a power bank. So in this case now, the what was I even explaining? Okay, I was talking about incomplete dominance. So they said in the case of Monohybrid jalapa, the the offspring, the offspring, you know, have uh, a kind of trait that is different from the two different parents that cross. The two different parents. That means they are neither this or this, even though they are intermediate. So let me use a cross to explain this. Let me use a cross to explain this. If we have a red flower, a red colored plant, you know, uh, a red colored Mirabilly jalapa, the 4 o'clock plant, crossing, being crossed with the white colored one. So this has red color. This has white color. So in this case, then at the end, it is have to cover that this is what we get in the first filial generation. This is what we get in the first filial generation. That means that they produce an offspring eh, that is completely different, phenotypically speaking, from the two parents. They are completely different. So this one now has a pink color. A pink color. You can see having a pink color. But then, if they are allowed to self-cross, if they are allowed to self-cross, we'll have this, 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 we'll have this. So this will now give us 1:2:1. Because you can't say 3:1 because this one is red color. The red color is still there, the pink color, and this is the white color. The white color. So this has distorted or altered the Mendelian segregation ratio of 3:1 and has turned it or has turned it into 1:2:1. So this is what we call incomplete dominance. Incomplete dominance. So the pink color, as you can see, is different from the white. It's also different from uh, the red color, even though it's also an intermediate. It's also an intermediate. Some people have tried to provide solution why this thing might have occurred, and some of them said, even though it's not scientifically approved, but then some people said, okay, there are two different uh, alleles, or two different genes are responsible for producing the red color. But since this one has only one of the gene, that the only color that is close to red is pink, and that is why you can see it having pink color. And some others say that some others now said that the mixing of the red and the white color now produce a pink color, just like you have a white paint. Sorry, the mixing of the red and the pink color now produce a pink. The mixing of the red and the white now produce the pink color, just like you have a white paint and a red paint. Now, by the time you mix them together, it will produce a pink paint. That is something that is closer to red. But well, like I said, these are not scientifically proved. All we know is that in incomplete dominance, the F2 offsprings will now exhibit a characteristics or a character that is different from the two parents that cross to produce it or to produce them. So that is it about incomplete dominance. Then the next one is multiple allelism. Multiple allelism. Multiple allelism. So in the case of multiple allelism, you will now discover that all this while, what we have discussed or Mendel discussed told us that we have discussed characters. Just looking at the time. 109. Okay, all this while we have discussed characters. We have discussed characters whereby uh, a single gene is located is located at a definite locus or loci, as the case may be. But then these modern genetics or researches done now discover that it's possible for us to have more than a pair of gene on a particular gene locus. So Mendel said that this is what we should have. Hmm? This is what we should have. So that means that we should have this, we should have this, or capital T, small letter t. That means that a single gene, a single gene is supposed to be found on a gene locus. But then modern genetics has discovered that there are situations whereby we have more than a pair of gene on a gene locus. And the situation whereby or the phenomenon whereby we have more than a pair of gene on a gene locus is now what we call multiple allelism. Can you see the word multiple? Multiple allelism or codominance is a genetic situation or a situation whereby there are more than a pair of allele on a particular gene locus. So you can have this. For instance, this can be seen in blood group. And in the case of blood group, you discover that A, B, and O are co-dominant. That means that these alleles are expressed within the heterozygous. The two are expressed. The two are expressed. There's no one. Sorry, not the two. I was trying to explain incomplete dominance. So sorry, what I'm trying to say that we have a typical example of multiple allelism or codominance, and that is the human blood group. The human blood group will have three different alleles. We have allele A, we have allele B, and we have allele O. So these alleles are all located on a particular gene locus. They are all located on a particular gene locus. So that is why you can now see we are calling it multiple allelism, where we have more than a pair of allele on a particular gene locus. So that's it about incomplete. That's it about multiple allelism. And of course, you should also know that mating between A and BB will produce individual AB. Will produce individual AB, and this individual AB are a kind of intermediate of A and B. All right. Another one is lethality. Another one is lethality. Lethality. Now, apart from distorting the Mendelian uh, the Mendelian segregation ratio, some genes also affect the viability of the organisms that contain them. And these genes are said to be leather genes. Leather genes. Leather genes are genes that are so uh, deleterious, genes that a kind of become so harmful in such a way that it does not even permit the birthing of an organism. When an organism has dominant forms of the gene, it becomes so it becomes so dangerous, or it becomes so harmful in such a way that the organism goes out. The nature by itself will now decide to terminate the organism, and that is what we call the spontaneous abortion. Spontaneous abortion. Or have induced abortion, or have spontaneous abortion or miscarriage. So nature by itself decides to terminate this pregnancy. in uh, in creepers, in chicken, you can see this. Then in human, also have a typical example like the polydactyly. The polydactyly, the presence of extra finger. So if somebody has extra finger, that person has this kind of gene constitution, and assuming the person marries another person with extra finger. When I say extra finger, I mean addition of one more finger apart from this normal 5 digits, and that is what we call polydactyly. Polydactyly. So if somebody has uh, extra five fingers with another person that has extra 5 fingers, when they marry, they're going to produce something like this: capital letter, then this. So nature terminates this pregnancy or eliminates this individual. And once this individual is eliminated, we now have this one. This one is still polydactyly. Then the only one that is normal is one. So this will now give us 2:1. Can you see that this has negated the Mendelian segregation ratio of 3:1? This is another phenomenon, genetic phenomenon that was discovered by uh, uh, modern geneticists, which negated the discoveries or the principles of Mendel. Then another one is what they call pleiotropism. Pleiotropism, in the case of pleiotropism, you now discover that one single gene influences simultaneously, and when I say simultaneously, at the same time, influences many phenotypes. You can see this in sickle cell. In the case of sickle cell, you see that the sickle gene now causes the sickling of the hemoglobin, the uh, uh, abnormal red blood cell, you know, uh, clumping of the red blood cells in the in the vessels, and so on and so forth. So you can see that one single gene is influencing many characters. That is another uh, a genetic phenomenon that negated Mendelian principle. So it says pleiotropism is a situation whereby a single gene simultaneously influences more than one trait or phenotype of an organism. We have so many of them, like I said, abnormal hemoglobin, we have sickling, we have clumping of the red blood cells, they'll have destruction of red blood cells by the liver, leading to anemia, also have increased resistance to malaria. So all these are phenotypic effects that are caused by one single gene. One single gene. That is called pleiotropism. All right. I think we have done justice to this. We have done justice to this. Let me round off. Let's round off by talking about. Let's round off by talking about some chromosomal aberration. Some chromosomal aberration. Chromosomal aberration. Chromosomal aberration. Just a minute. So, what to look at chromosomal aberration. When something is aberrated, that means that is abnormal. So, we want to look at chromosomal aberration within the autosome and within the sex chromosome. The one that happens within the autosome and the one that happens within the sex chromosome. So, we're going to pick one from the autosome, and autosome are the of course chromosomes are in two types. We have the one found in the somatic cells, that is called the autosome. Then we now have the one found in the cell cells. That's what we call the sex chromosome. The sex chromosome, and of course these are the ones that determine the sex of an individual. So the first one we want to look at under autosomal aberration is the Down syndrome. The Down syndrome. The Down syndrome. The Down syndrome. So in the case of Down syndrome is a case of aneuploidy, or what we call trisomy. Eh? Trisomy. Trisomy. Instead of having 46 chromosome, the individual got another copy of chromosome, resulting to 47 chromosome. Mm? Resulting to 47 chromosome. So this is the case of aneuploidy or trisomy. Or trisomy. Mm? Trisomy. So what causes Down syndrome? Down syndrome, you have two main causes of Down syndrome. The first one is what we call the non-disjunction non-disjunction of chromosome 21. Mm? Chromosome 21. Chromosome 21 is the smallest chromosome in the karyotype. Mm? The outline of all the chromosomes, the one that has the smallest shape, is chromosome 21. But it causes Down syndrome. When the chromosomes are not well separated, and don't forget that the primary oocyte of a female child or of a female begins at the fetal stage or within the fetal period of life. That is when the primary oocyte. And the this primary oocyte, of course, go into meiosis, which is never completed. Mm? They go into meiosis and they remain at prophase 1 of meiosis until the lady gets to the age of puberty. And once the lady gets to the age of puberty, then of course the meiosis 1, is completed, and within the period it now transforms into the secondary oocyte. That is during ovulation. That is when that first meiotic life cycle is completed. And when it's completed, the primary oocyte, a kind of graduates or transforms into the secondary oocyte. This secondary oocyte will now go into another the second phase of meiosis. We discuss meiosis. We have meiosis has first phase and it has second phase. And this second phase of meiosis, is also arrested at metaphase 2, and it remains there. It remains there until when that individual is ready for fertilization or is ready for pregnancy. So meiosis 2 is not completed until during fertilization. It is during fertilization, just before the two pronuclei fuse together. The two pronuclei. When the sperm breaks through the zona pellucida, then it loosens up and transforms into what is called the pronuclei, the pronucleus. Then the nucleus of the egg also, enlarges to also form pronuclei. So within the period of the penetration of the sperm, but before the fusion of the sperm nucleus with the egg nucleus, that is when this second meiosis is completed. So you can now see that this non-disjunction of chromosome 21, is a very big issue, because the spindle fibers would have been weak. mm, the spindle fibers would have been so weak that it will not allow the normal separation to occur. So you now get a child having 47 chromosomes instead of 46. So this is what imparts the effects of Down syndrome. And of course, Down syndrome is common to women that are older. mm. It's common to women that are older. The higher the age of a woman, then the higher the probability that that woman will not birth a child with Down syndrome. So it's discovered that women at the age of late 40s are more susceptible than women at the age of 30. And that is how it continues. And I've told you the reason. Then another second genetic reason why Down syndrome may occur is as a result of translocation. There's a kind of translocation. Segments of chromosome 21 are broken and rejoin that of chromosome 14. This translocation of chromosome that occurs between chromosome 21 and chromosome 14 can also result to Down syndrome. Then people who have this genetic condition, of course, they have they're usually short and mental retardation, even though some end up producing offsprings, while some do not. But it brings about mental retardation and so on and so forth. So that is it about Down syndrome. Down syndrome. Okay. Then the next one is Klinefelter syndrome. Klinefelter syndrome. In the case of Klinefelter syndrome, this one this one is no longer we have left a chromosomal aberration or autosomal chromosomal aberration within the autosome. Chromosomal aberration that occurs within the autosome. We now want to go into the one that occurs within the sex chromosome. The one that occurs within the sex chromosome. So in the case of the sex chromosome, we have the Klinefelter syndrome. The Klinefelter syndrome. The Klinefelter syndrome. So this is also another case of aneuploidy, and it happens in males instead of having SY. Of course, you know that a male should have SY as the chromosomal constitution, whereas a female should have SS. So instead of a man having SY, the man now has SSY. SSY. So in this case now, you see that the man begin to have a broadened hips, just like a woman, and the development of breasts, then high voice, and there's some other feminine characteristics. Feminine characteristics you observe in such a man. A minute. And some other feminine characteristics you observe in such a man. So when it happens, that is the case of Klinefelter syndrome. We also have other mosaics. Other mosaics like SSS, SSY, SSY, also having some of them having SSY and what have you. Okay, so that is it about Klinefelter syndrome. And of course, you should know that this is not common. This is not common. It happens in one out of 1,000 childbirths. Another one is the one they call the SYY, the SYY males. Now, this is another issue. You may not say, okay, the extra Y, does it cause any problem? Yes, it does cause problem. So, in the case of SYY males, instead of having normal SY, they now have SYY. And we have two different conditions or two different characters or characteristics that are exhibited by this kind of people. Number one, that they have below average intelligence. You know, mentally they are not mentally stable. And apart from the fact that they're not mentally stable, you can also discover that they also grow above average, more than about 6 feet tall or more than 6 feet tall. And people have always linked this genetic constitution with violence, because some scientists in the past performed experiments and conducted research, and they discovered that majority of the men in prison, have this genetic constitution. But then some people in so many countries have tried to use this as an excuse to be exonerated from the crime they committed. But then the juries were wise because having this genetic constitution is not a reason why the person should be predisposed to violence. It's not a reason why the person should, go into or be violent in nature. It doesn't. But then, just like I said, most of the people in prison, after the research, it was found that a majority of them have this genetic constitution. But geneticists are still saying that it's not enough reason why one should be violent. Then the last one is Turner syndrome. Turner syndrome. In the case of Turner syndrome, you see that a lady or yes, a woman, instead of having S1, SS, now has S0. So instead of having two S chromosomes, the person now have one S chromosome. So this is monosomy. This is monosomy. Monosomy. Monoso, monosomy. Monosomy. Of course, in the case of trisomy, we'll have extra one. In the case of monosomy, we'll now have 46 minus 1, giving 45. Giving 45. So this, you see that these are women out of 5,000 childbirths. These kind of women are not usually fertile. They're not usually fertile, and they also fail to menstruate. They fail to menstruate and fail to produce normal secondary sexual characteristics that you see in females. Some of them have short stature and will have folds of skin around their neck, folds of skin around their neck, and some of them are mentally normal, or mentally they are usually normal, except that they are frequently weak in mathematics and some spatial perception, even though they may even though they may even though they may be mentally normal, but uh, they have issue with uh, mathematics, and some other hard concepts that they find it difficult to comprehend. All right, we are gradually coming to the end of this class. Let's now quickly, before we say bye-bye to this class, uh, let's now quickly talk about selected hereditary metabolic disease. We have some selected hereditary metabolic diseases. I want to mention some of them. I want to mention some of them. I want to mention some of them. After which, let's mention some of them. Some common hereditary metabolic diseases. The first one is albinism. Albinism. Albinism is common, and of course, we have two major types of this albinism. We have the one they call the ocular albinism, and we have the ocular cutaneous albinism. The first one, the ocular albinism, is caused by genes that are S-linked. We call them the S-linked genes. We have genes that are found within the S chromosome, the S-linked genes. These ones are the ones that cause the ocular. They will now have the one they call the ocular cutaneous albinism. Ocular cutaneous oculo. Okay, this is ocular. Okay, oculocutaneous albinism. So this one is not caused by autosomal recessive traits. Is a kind of autosomal recessive trait. Eh? That means that it's caused by genes that are found within the the autosomes. Genes that are found within the autosomes. That is where you see mutation within this gene can result to this second type of albinism. this second type of albinism, which is called the oculocutaneous albinism. That of ocular, like I said, you is an X-linked recessive trait. Mm, is an X-linked recessive trait, and you can uh, you can see some of the clinical manifestations. Some of the clinical manifestations for this ocular albinism, something like a photophobia. Photophobia. A person begins to usually express discomfort in bright light. Then one they call the nystagmus. Nystagmus, uncontrolled movement of the eye, upwards, sideways. That is what we call nystagmus. This can also be a condition that somebody with this ocular albinism will experience. Also have absence of binocular vision, inability of the 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 eyes to uh, see clear vision, to see clear vision. Then this is usually, remember the word ocular has to do with the eyes. So this kind of albinism, all the effects, all the clinical manifestations are talking about the eyes. Then we have, in addition to these clinical manifestations that have to do with the eyes, the oculocutaneous albinism also has additional clinical manifestations, such as what we call keratosis, solar keratosis. We have solar keratosis, solar keratosis. We have solar keratosis. These are the clinical symptoms for the second type of albinism, which is the oculocutaneous solar keratosis. Solar keratosis. Then, of course, we have the basal cell carcinoma, the basal cell carcinoma. Then we have the squamous cell carcinoma too. The squamous cell carcinoma. The squamous cell carcinoma. So have solar keratosis when you have patches or scales on the skin, a kind of lesion, especially when exposed to sun. That is why you have solar keratosis. They'll have a basal cell carcinoma. Anytime you have carcinoma, you know that is a kind of a type is cancer. This is a type of cancer that is found within the basal cell of the epidermis. Then within the squamous cell, the squamous cell. Of course, these are different types of tissues, different types of tissues, and that is epithelial tissue. Different types of epithelial tissue. Eh? Especially the squamous cell, the type of epithelial tissue. Okay, so this is a type of cancer you see within the basal cell. This is also the kind of cancer you see within the squamous cell. So these are some of the clinical manifestations of the oculocutaneous albinism. Then another one is what we call alcaptonia. Alcaptonuria. Alcapto. So when we talk about, remember what we have been discussing is selected hereditary metabolic diseases. selected hereditary metabolic diseases. So when we talk about alkaptonuria, is a rare autosomal recessive disease. Mhm. So this is another autosomal recessive disease, and this disease is as a result of deficiency or mutation in the gene that is called the homogentic oxidase. Yes, homogentic oxidase. So this homogentic oxidase, if there is a mutation or a defect or a deficiency in the function of this gene, then it becomes a problem. It becomes a problem in the sense that it will prevent the uh, it will prevent the normal conversion of phenylalanine and tyrosine. So, since this enzyme is necessary in the intermediate metabolism of phenylalanine, the homogeneity increases. So, during the metabolism, oh yes, that's what I wanted to say. During the metabolism of tyrosine, tyrosine is an amino acid, and phenylalanine is also an amino acid. So during the normal metabolism, one of the precursor compound that is found is the homogentic acid. Homogentic acid during the normal metabolism breakdown of tyrosine and phenylalanine. Then this gentic acid, homogentic acid is not oxidase now. I'm talking about the acid. Is usually produced. But then there's this enzyme that is called homogentic oxidase that helps in the breakdown of these acids. But if there's a deficiency in this enzyme called homogentic acid, that means that it will not be able to perform its function of breaking down the acid. And that means that the acid will begin to accumulate. And once it accumulates, it begins to result to many clinical manifestations, such as arthritis, that is similar to the one of rheumatoid arthritis, and also uh, black patches on the skin, you know, and also the person having a kind of dark urine. The person having a kind of dark urine. Okay, dark urine. So these are some of the clinical manifestations that one may experience if the person has this condition, alkaptonuria. 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 Remember, it's basically it's basically results to the accumulation of this acid, homogentic acid, because of deficiency in the function of the enzyme homogentic oxidase, which is supposed to break down the acid. Okay, then phenylketonuria. Phenylketonuria is another condition. Another condition, of course, you see this mainly in babies. Usually, if the level of phenylalanine in the body becomes high, then there's an enzyme that is called phenylalanine hydroxylase. So, if the level of alanine in the body becomes high, then the body naturally converts excess phenylalanine into tyrosine. Tyrosine, because excess of phenylalanine can cause issues within the cells or within the body. So, this enzyme phenylalanine hydroxylase helps in the conversion of excess phenylalanine into what's it called again? Into tyrosine. Into tyrosine. So, but if there's an issue or mutation in the gene that codes for this enzyme, and this enzyme is not available or does not work well or does not function well, then of course that conversion will not occur, and the level of phenylalanine will accumulate. And this accumulation of phenylalanine can cause